Hello everyone, welcome to 10-Minute Physiology. In today's video we're going to talk about smell, so with that let's give it a go. So I would like to begin with the general description of our olfactory sense. So what we have here is inside our nose we have the olfactory epithelium. So the olfactory epithelia is basically where all our smells are going to be detected. And protruding through the olfactory epithelium, we have our odorant receptors. So the odorant receptors, which we see right here, are actually neurons that turn over every 4 to 8 weeks. This means that these neurons actually die and are replaced every 4 to 8 weeks, which is actually quite interesting for neurons since they usually don't do that. So we have our odorant receptors sticking through the olfactory epithelium. And then what we also have here is that on top of these odorant receptors, we have a layer of mucus. Now this mucus layer that rests on top of the olfactory receptors are basically going to contain a few different things. So the first thing are the odorants themselves. So these are molecules that when they bind to the odorant receptors can cause a signal and cause us to have the sensation of smell. We can also get odorant chaperones, which are proteins that can carry odorants and bring them to the receptors. We can also have enzymes that basically break down odorants to keep them from blocking receptors for too long. And we can also have antibodies for the immune response. So we have these receptors that are bathed in mucus, and then they produce a signal when they bind to their odorants and bring them to the olfactory bulb. So another thing that you see here is that inside the olfactory bulb you have these mitral cells. And these mitral cells are basically going to synapse with a number of different receptors, cells. So the olfactory receptors synapse onto one mitral cell, as we see right here. And these mitral cells will then project their axons through the cranial nerve number 1 to the brain. So it's important to realize that these olfactory receptor cells are going to be bound or synapsed to different mitral cells, but the brain maps them in specific ways so it can see what we are actually smelling. So now that we have a general idea of the olfactory sense, let's actually get a closer look at how the signal transduction pathway occurs. So this right here is a close-up of an olfactory receptor cell. And what we see here is that the olfactory receptor cell has a receptor cilia or microvilli, and it also has a cell body as well as an axon. So now what we're going to do is we're going to see what the signal transduction pathway is for these cells. So in order to do that, we're going to get a close-up of the microvilli, and we're also going to get a close-up at the receptor. So this right here is a receptor for a specific odorant. And when the odorant binds to this receptor is going to initiate a signal transduction pathway. So the receptor in this case is going to be coupled to a G-olf protein. So this is a G protein that is in the olfactory system. So when we have this G-olf protein activated, it's going to move to adenylate cyclase 3 and it will activate it, causing it to produce cyclic AMP from ATP. Now the cyclic AMP is going to interact with the CNG channel, which is a cyclic nucleotide gated channel. So when cyclic AMP binds to it, it causes it to open, allowing sodium and calcium to flow into the cell. Now the sodium and calcium not only produce a depolarization, but the calcium is also going to interact with another channel, which will allow chloride to leave the cell. All of these effects will lead to a depolarization, which will then move up the cell, producing specific effects. So it's important to realize that each cell has its own class of odorant receptor. However, the same signaling mechanism is going to be shared by all odorant receptors. So in other words, it's univariant. So therefore, the brain will identify what we're smelling based upon mapping. So the mapping is going to occur in the brain, and it's going to allow us to see the identity of our smell. So now that we have an idea of the signal transduction pathway, let's take a look at the electrical activity within this receptor cell, and then we'll call it a day for this video. So the last thing that we're going to look at is what the electrical activity looks like in these cells. So when you have your odorant receptor stimulated, the depolarization climbs up the microvilli towards the cell body. And these depolarizations that are produced in the microvilli are going to be the receptor potentials or graded potentials, as we see right here. Now, as the signal moves up the cell, it's going to go to the cell body. And in the cell body, we see something like this, where we have a receptor potential, and on top of it, we have these spikes. So these spikes are going to be the action potentials. And then as we move further up into the axon, what we see is only action potentials. So it's important to realize that the stimulus intensity is going to be coded in the firing frequency of action potentials. So the greater the stimulus intensity, the greater the firing frequency. So I hope this video helped you have a basic understanding of how we smell things, and I hope to see you in the next one. Thank you for watching, and I hope to see you next time.